think we got it. This. First things first, quiz went live. I think I set the due date for Tuesday. End of the day at Tuesday. Covers the terminology stuff. All right, you got 15 minutes to do it. 15 minutes to do that quiz, so should have plenty of time. Make sure you review your notes. Study your notes ahead of time. Those of you that haven't had me before, there are some questions that are multiple answer type questions, where you might ask a question, let's say, maybe I diagram the life cycle, have a picture up like we finished, and I'd say, which of these hosts are intermediate hosts? And the hosts are labeled like A, B, C, D, or E, or something like that. So it's not just one answer. We have multiple intermediate hosts on those life cycles. You have to select both of them. All right. Catch, though, is on these Blackboard questions, it's could be all or none. All right. So if you didn't pick all of them, then you lose the entire point. I don't do that. What I do is for each one you select, you get partial credit for that answer. If you select an incorrect answer, then you lose a quarter of a point. So if that multiple answer has 2 answers that have to be selected, each one gets you half a point. If you select one that shouldn't have been marked, then it's a quarter of a point deduction. In no way can you ever go below 0. So if you pick like the 2 wrong answers, that's a total of negative 0.5. You're not going to get negative 0.5 for that question. It's just a 0 out of 1. And on these questions, for the most part, they're all worth 1 point. You may see some of these questions, like for the quiz, they're all worth 1 point. But you may see some questions in future quizzes or on exams where that question is actually worth 2 points, because it may be, I may ask a question that's say, which of these mechanisms is a parasite evasion mechanism? Actually, bad question. Which of these adaptations increase parasite transmission? And there may be 8 different answers, 8 different options. So with a question that big, I might assign it 2 points instead of just 1 point. On this one, we don't have any true false. So it's going to be a mix of multiple choice or multiple answer. I think for this first question, or for this first quiz, I did state, choose all that apply for the multiple answer questions. I think I got all of them. But in future ones, I won't state that, because when you take the quiz, you'll know. It's either check boxes or it's what they call the radio button. You can only pick one answer. We don't have any fill in the blank on these. So that's due on Tuesday by 11:30 at night. All right. If you miss it at 11:30, you can still take it. Just remember, we get a late penalty attached to it. And I have it set up so it'll only show you the score. And then what we'll do is, as we get closer to the exam, so like the week of the exam, or if everyone goes through and takes it and is done with it, then what I'll do is go back and release the actual key, so you can see what you missed, what the correct answers were, or what the incorrect answers were. Right? If you didn't do it, there has to be some point. You won't have access to that, because in order for you to get access to that key, you have to have a grade in the Blackboard grade book. So one thing that I kind of found is, if I automatically give you a 0, you can't go in and retake it. So take the quiz, get access to the key. That's up. Next up, Parasite Species presentations. I had 2 people reach out to me with their picks. So Gordius is taken and Gyrocotyl is taken. So what species do you want to do? Who wants to speak up first?
Gordius and what was taken?
Gordius and Gyrocotyl. It's under Cestodia.
My first choice was Myxosporia.
Yeah, Myxobolus.
Yes. Sorry.
Yep, Myxobolus.
Mine was Pediculus humanus.
Pediculus. Okay.
I don't know if I'm gonna say this right, but mine was Babesia.
Oh, Babesia.
Yeah.
Oh, hold on. Babesia. I'm still learning names.
Amber.
Amber on Babesia. All right.
I'm Shania. I struggle with this. It's the strong.
Strongyloides.
Yes, thank you so much for that. Skylar,
Malaria. All right.
I'm Emily.
Hold on.
I'm gonna choose the Exalius test, the one that the parasite didn't finish.
Phagocytosis.
Yeah.
Okay. Name.
Nicole. The Dario Define Bruno.
Back to find me. All right. Name.
Emily.
Also do the
All right.
Alexia for Hymenoleuctus.
Analysis. Alexia.
Yes.
Okay.
Paula, I went to the Capillarium.
Oh, capillaria.
Yeah.
Right.
I'm Jenna. I guess I'll just do the legs. The
Ridiculous.
yes.
Hope already picked that.
Oh, she did.
Yep.
Sorry. I will do. Did anyone take the stercoralis?
Strongyloides. Yep. She did.
Okay. What in the maxillopoda did anyone take?
Phagocytosis.
Okay, I'll do the other one.
Stachylina.
Yes. Did anybody do cryptosporidium?
No.
Okay.
Rivaroia. It's it. Toxicara. Kyle just took that. Yep.
The one at the bottom.
Anasagus.
Is a Campylobacterium taken?
You got it. Capillaria. Paula has it.
someone said they put it in the chat.
Oh, is there? I didn't see it.
YouTube.
Elliot. All three of those are taken. Is there a timestamp on that? I can report the user. I should report Elliot. There's no timestamp on that. No, all three of them.
Is there anything left in Acanthocytoma, that category?
Yeah, all three.
I'll take the first one.
Maniliformis?
Yeah.
All right. Elliot doesn't mind doing another one. Who else is not? Cole? Pick one. All right. So it looks like either platyhelminths, nematoda, or acanthes. Prophyllocalis? No. You can have that. All right. All right, just Neil. Got one?
I don't know the one that I'm not taking.
So all of the ones on the right are taken. So you've got the monogenes, both monogenes are available. Of the trematodes, we have Uvolifer, Megalodiscus, and Spidigaster that are available. Cestodes, you have Ligula. For the nematodes, you've got Eustrongylides and Huffmanella, Haemonchus and Balis ascaris.
I'll take Ligula.
Ligula? All right. All right, so what I'll do is post the assignments, update the form and post it. So Michaela, Janelle, Alexia, Gabriella, and Chetta might not be. Oh, hold on, Elliot. Saculine is taken. Elliot.
Nice meeting you, Steve.
Let's see here. How? Guess we're just Elliot's the last one. So, what I was gonna say, Michaela, Janelle, Alexia, Gabriel, and Chad. And now Elliot. Elliot, that's good. You got Uvolifer. All right, so we've got 6 people that are going to present. They're going to present on February 18th, 15th, or the 22nd. Look at your schedules. See what day you'd like to present. I would prefer not to do the 22nd, so that way we have that entire week. Entire week available to ask questions. If you remember, the February 22nd, that Friday, is our lecture exam, and then our lab practical is the following. If that's the only date you can go, that's the only date you can go. These presentations will be about 10 minutes, say 7 to 10 minutes long. You're going to introduce the parasite, talk about the taxonomy, classification scheme, give the life cycle, find pictures, images. You may need to download PDFs that have the diagrams, maybe the species descriptions in it. All right. With the life cycle, it's pretty good to draw it out. So you can use, the boxes and the arrows on PowerPoint, or you can draw it out by hand, scan it, and insert it into the presentation. All right. I don't expect anyone to have, like the graphical quality life cycles like we see in our textbook. All right. As you go through it, you will note some interesting things tied to it. Either an interesting life cycle, maybe because of the types of organisms that they infect. Maybe it's where they infect those organisms. Maybe it's behavioral changes that they induce on those life cycles. Maybe it's the life cycle stage that's like really strange, or how they reproduce that's pretty unique. Or it could be pathology. Maybe they cause a specific pathology that is very common, and common enough to where it has an actual name, like a common name to it. So we can look at this stuff. And we could say, yeah, it's trematodiasis, right? That's vague, but if you have Chinese liver fluke, yeah, that's a quality common name, all right? Something like that. Introduce the class to these parasites. Next week in lab, I'll ask you to pick your dates, all right? Any questions? So what are we doing? So this presentation is posted on Blackboard, or at least the PDF of the presentation is posted on Blackboard. So you can download it, use it, take notes on it. This will be the only one that you get because this presentation is more of an FYI type presentation. We're going to see a lot of this material later on in the class when we talk about specific parasites. We'll come back and talk more specifically about some of the immunological responses, what works, what doesn't work for in terms of fighting the parasites. But I don't want to just throw that stuff out there and you not understand, or at least you haven't seen, just the broad overview of our immunological response, or our system. So this is what we're going to do. A brief introduction. I'm not going to ask questions. We won't have a quiz tied to this. So I just kind of start off with, a classic, what I would call a classic Star Wars quote, the good Star Wars. Prequels are okay.
You're one of the purists.
The latest ones, I don't know. Force Awakens, I enjoyed it, but then I just felt like it dropped. I was a little disappointed. Which I guess is why they're gonna remake it on Disney Plus. Did you see that?
Like
Redo the sequel trilogy, 1 season for each, estimated 9 to 10 episodes.
I'll be honest.
Are you kidding?
I'm actually kind of excited about the thought of that.
You know what it is? It's money. That's what it is. We can remake it and get even
That's
more.
what Disney does.
All right, so in the classic one, the Millennium Falcon gets away. That guy says, and our stormtroopers were outfought. Our Starfleet evaded. How is that possible? Whose ship was it? That's difficult to say. She has false identification markings and a forged registration. Moreover, she was an extremely fast and elusive vessel, probably piloted by one of the smugglers who congregate in that region. How is this applicable to parasitology? Well, one of our evasion mechanisms could be to hide yourself from the immune system, make you seem like you're something different. So then the host, if it doesn't notice you as foreign, it ignores you, and thus you can survive. So it's very applicable, and we'll talk about at least one parasite that does this. All right, so some of the terms for immunology, just quickly, host specificity. All right, we've talked about it in terms of like generalist versus specialist parasites. And one of the questions you could ask is, what determines that? What determines the specificity? What determines if you're a generalist, or what determines if you're a specialist? Well, it's two different things. One is the host itself. All right, how successful is the host in eliminating you from their body? All right, if you have a host that has a very good immune response that is able to detect and eliminate you, that host probably isn't going to be a host. All right. The other side is the parasite. How successful is a parasite at evading the host immune system? Now, the other aspect of the parasite is, did the parasite obtain or receive the correct cues that signals it needs to start maturing, starts developing? All right. So for this presentation, we're talking about immune defense versus immune evasion. Pathology, morbidity. What are those terms? Those terms are all about, the negative consequences, the negative effects that are exhibited by the host. Now, usually, these are all directly related to the host defense mechanism. Yeah, the parasite can cause some mechanical damage and physical damage, but a lot of the pathology that we end up seeing is tied to host defense mechanisms. Example of COVID is the loss of smell. All right, the virus didn't damage those. It's suggested now that it's actually our immune response. The immune response ends up in the death of those sensory cells. That's a consequence of the host defense mechanism. Susceptible versus resistant, that's in terms of the host. The opposite terms for the parasite would be infective versus non-infective. All right, so in terms of the host, you can be susceptible or you can be resistant. A susceptible host is one that can become infected with the parasite. The resistant host is one whose physiological status prevents the establishment and survival of the parasite. All right, and we'll talk about those a little bit in more detail. Infective versus non-infective, these are equivalent terms. If you're infective, you can successfully infect and establish inside that host. If you're non-infective, you can't. All right. In terms of human disease, we often you'll see the term infectious. All right, just kind of means the same thing. All right. What determines infective versus non-infective? A lot of times it's maturation of that parasite. Are we at the appropriate stage where we are now infective to the host, where we can now detect the symptoms and or survive the initial infection process and then develop to the next stage? So, resistance and susceptibility. Well, resistance is more of a specific term. And that is you have some sort of physiological mechanisms that's preventing the establishment of the parasite. Perhaps it is our gut, strong stomach acid, low pH. That could be a physiological characteristic that gives us resistant to some of these trophically transmitted parasites. Flip side, and a related term, is immunity. Now, what's different is immunity is resistance to the parasite. You can't get infected or you're highly resistant to the infection, but it's not due to the physiological status. It's actually due to your immune system. It's due to your memory response specifically. All right, so you have cells and tissues that can recognize and then protect the animal against this non-invader. Sometimes it is that memory response. It's providing protection. Other times it's our innate response that actually kicks in high gear to really prevent the establishment of the infection. All right, so we're going to see both. We'll probably use immunity more than resistance, but resistance becomes an important predictor as to which host can serve as an adequate host, or which free-living organisms can serve as an adequate host. Now, in terms of immunity, what are some of the pathways that would confer immunity? What are the types of immunities? We've got innate immunity. All right, we have adaptive immunity. So innate immunity is just our built-in response. We don't have to see anything previously. It kicks in and tries to recognize those foreign invaders. Adaptive immunity is also that acquired resistance. That's we've seen the antigen before. We've built a memory response, so if we see it again, we're really fighting strong. Then we have premonition and concomitant immunity. These two are somewhat related, slightly different process, but both of these are tied to the parasite. Both of these are tied to the parasite. And what it is you're infected with the parasite's doing fine, but because the immune system is fighting that one parasite, any new invader that comes in is wiped out. So it's kind of thought that these two things, premonition, concomitant immunity, are some ways to prevent overinfection of that host. And we'll talk about these. We have different examples of this premonition and concomitant immunity. All right, so innate immunity is that mechanism of defense, as I just said, it doesn't depend on prior exposure. I guess we already went over here. I already did those. So premonition and concomitant immunity, we will see these. I will define these again. But premonition is resistance to reinfection or superinfection conferred by the still existing parasite. And what's important about this one is that our parasite is harmed. The immune system is being successful about that parasite, or against that parasite, but the parasite is replicating. So is replicating faster than the host is removing them, or replicating at the same rate as they're being removed, so that infection persists, but any new infection is stopped. In concomitant immunity, our parasite that's in there is completely unaffected. It just says, hey, whatever you do, throw me everything you've got. So that parasite is unharmed, but the benefit is that our host is now resistant, or I should say, exhibiting immunity to other stages. In some cases, it's other parasites. Sometimes it's bacterial and viral infections too. You may have heard that if you have one type of, let's say, viral infection, you might be more susceptible to a bacterial infection. Or if you have worms, you're more susceptible to these different types of infections. And again, we'll point those things out as we get to them.
Can you go back? Sorry.
As I said, we'll see these terms again. So you don't have to worry about memorizing them.
So how often do you see like a sort of overreaction of the immune system as far as parasites?
You can see it. it's gonna be pretty rare. If you see it, it's usually something happened to that parasite, thus releasing excess antigen, and then you have this systemic shock that sets. That's a characteristic of hydatid disease, one of the dangers associated with it. Yeah, but like, as with parasites, I mean, if the host overreacts and kills itself, that's a bad thing for the parasite, unless the parasite's really good at replicating and getting out quickly. So typically, if you cause a lot of pathology, a lot of morbidity, you're probably more infectious. Kind of that's our trade-off. All right, innate defense mechanisms. These are things that you don't have to see it before. If you got several different things, some of these things you probably didn't even consider. So unbroken surface of our skin, that's a first line of defense. Nothing's going to get through it. Or mostly nothing's going to get through it unless it burrows its way through. All right, soft outer surfaces usually have a mucus layer, and that mucus layer is actually acting as that first line of defense. In order to get through it, that mucus layer, in order to get inside the host, they have to traverse that mucus layer. Kind of hard for viruses, kind of hard for bacteria to do that. We also have these mucin in the GI tract. All right, so our gut also has this mucus layer, also acting kind of like as a force field.
Is that why for the coronavirus they test like your nostrils for the mucus layer or mucus membrane?
Now they're releasing the virus into that mucus. The cells are replicating and being released in that, and then when we sneeze, a lot of that mucus comes out. Do slow motion sneezing videos. You could probably spend an hour killing time on YouTube doing that. So mucus layers, mucin layers. We didn't even talk about immune molecules in the mucus itself. So you're going to have antibodies that could be in there, acting as protection. You have and as well as these antimicrobial molecules. So protein pattern recognition receptors, PRRs. They're going to be on the surface of cells. So if they recognize, an invader that has one of the epitopes, they're going to tag them, and then you've got your immune system that comes in and takes them out. But they can also be secreted, so they're in the mucus layer, or even possibly in our blood, that when they bind, then something else comes along, kind of touches it, you've got a slight binding, and then recognizes that tag is on another cell, and that starts our cascade. All right, toll-like receptors, that's kind of what they are. That's what they do. When a toll-like receptor binds, it's going to activate the innate immunity and then start the adaptive immunity process. Pathogen-associated molecular patterns. These are what we recognize. That's what our innate immune system is recognizing. Those are things that are going to be on the bacteria. All right, and then you have this complement cascade. So this is another type of innate response that you get binding, get recognition of that foreign invader, and then all of a sudden you start expressing complement that comes in, and what I'll say is start to poke holes in the membrane of that invader. All right, you didn't have to see it before. I'd like to say non-specific, but it is specific. It's going to recognize that foreign invader, and then it's going to hit with complement. Complement's an important defense mechanism against parasites, especially some of our protozoan parasites. And then we also have other chemical defenses like pH, hydrolytic enzymes, parasitological substances, so immunoglobulin A and lysozymes. Those are important innate immune defenses that'll help stop the parasite. So Trichomonas vaginalis, this flagellate parasite that we'll learn about in the other flagellate section, lives in reproductive tract, and in vagina it's not conducive for it. So when it gets in there, it actually adjusts the pH so that it is much more habitable. So pH is an innate chemical defense. Now, as part of this innate defense, what ends up happening is phagocytosis. So the innate immune system recognizes the foreign invader and then calls in other cells like macrophages that will come in and bind and chew up our invader. So this is just kind of an example. And I don't know where I pulled it from. Maybe Wikipedia, maybe from like a free textbook or something like that. Our bacterium comes in, gets recognized, gets tagged, and then our macrophage comes in, has receptors, binds to the bacteria, and then phagocytizes them. All right, so endocytosis happens. You get that now becomes a phagosome. Phagosome fuses with the lysosomes. You've got changes in the pH that happens into this phagolysosome. And now you have breakup and degradation of that bacterial molecule. All right, it doesn't just stay there. Our macrophage is then going to export it out of the cell as soluble debris. This is cellular defense, and this can work. But it can only work so well because this is against a bacterium that's pretty small. How do you think it's going to work against a large worm? Right, it's not going to be able to engulf the entire worm, so we're going to need some other mechanisms. What cells take part in this phagocytosis? Well, this occurs in both vertebrates and invertebrates. So our snail hosts, they get infected, they have innate cells that will take out some of these foreign invaders. The name of their immune cells tend to be associated with the group or with their classification. So the archaeocytes, the amoebocytes, the hemocytes, the celomocytes, etc. They all basically do the same thing. They function a lot like macrophages. All right, they function a lot like macrophages that we have in the vertebrates. So in our vertebrates, you can have several different cell types that can undergo this type of phagocytosis pathway. Right? We've got our monocytes. These are going to be cells that are part of the reticular endothelial system, the RE system. All right? The main one are the macrophages, and we're going to see it. We're going to see parasites that take advantage of this and will infect these cells. Maybe not macrophage, but perhaps these Kupffer cells in the liver, the microglial cells in the central nervous system. All right? Dendritic cells. They're also important phagocytosing cells. All right? The immature dendritic cells, definitely, they circulate, they're active. All right? And then you also have these granulocytes or polymorphonuclear leukocytes, the PMNs, that consists of our neutrophils, eosinophils, basophils, mast cells, and so forth. A lot of times you go get blood work, you're not feeling good. If they come back with a high eosinophil count, typically associated either with allergies or parasitic infection, because it's an innate response. And the innate response tends to be amplified when you get parasites. Incidentally, if you have a parasite and you have high eosinophilia, chances are you're probably not suffering from allergies. Your body's so focused on fighting that parasite that it ignores everything else. Did you talk about that? Who said immunology? Did you talk about that? Usually do. So, the adaptive immune response requires exposure to our foreign body. All right. So what's going to happen is we see this foreign antigen, abbreviated AG, an antigen, and then our body's going to produce a response to it. All right. A lot of times it starts with our innate response and then escalates and says, okay, we've got this innate response happening. Let's shuttle off some of these particles to form a memory response. All right. This memory response develops antibodies. And the antibodies are going to be around, and they're going to recognize that foreign invader. All right. Now, these antibodies could be free-floating, or they could be attached to cells. Right? And these would be on the surface of B cells. Now, if they're attached to B cells, and that cell, that antibody on the B cell binds to the antigen, then you're going to start a cascade to say, hey, I've seen it again. Let's ramp up all the antibodies. Let's ramp up antibody production again. So the antibodies can go bind. Once they bind, they tag that foreign invader, that tag the antigen, and then our T cells start to come in and start to try to take them out. All right? So T cell receptors are also part of our antibody response. These are going to be the immunoglobulins. So antibodies are immunoglobulins. They're going to be on the surface of our T cell, our T lymphocytes, and we've got Th1, Th2 cytotoxic T lymphocytes. We've got regulatory T lymphocytes, and so forth. All right. So how do you have these cells? How do they recognize self versus self, or self versus non-self? Because some of our cells get damaged, and our own body will go in and clean them out. All right. Now, if they went in and cleaned them out, it's possible then those macrophages start and induce a memory response. But we don't really want that, because the memory response then would be against our own cells. And then you'd have a ramped up autoimmune diseases, right? So how does it identify self versus non-self? It does so through these major histocompatibility complexes. These are proteins embedded on the cell surfaces that basically say, if we have it, it's self. If we don't, if there's not a match, it must be a foreign invader. Now, the class 1 MHCs are on all cells. The class 2 are on the immune cells. So our immune response is looking for all of those. The immunoglobulins are antibodies. They also have several types, and they're going to have different functions. Now, these are the abbreviations: IgM, IgG, IgA, IgE, IgE. All right, but they're actually named for the Greek letters. So it's Ig mu, Ig gamma, Ig alpha, Ig delta, or immunoglobulin delta, immunoglobulin epsilon, and so forth. And you'll see some of these different responses. I think I have them here in this presentation. So what are these functions of antibodies? Right? They're not just used for recognition of foreign invaders. All right? If they are, then that's opsonization. So the antibodies are going to be circulating. They recognize, they bind, and they basically coat that invader. And once it gets tagged, once it gets coated, then the macrophage is going to come in, bind to the bottom part, to the crystallization fraction of that antibody, and then start to engulf it. So our antibody, our antibodies take on, it's really crappy. Yeah, 2 heavy chains, 2 light chains. All right, and you've got your epitope regions that'll bind to your foreign invaders, and then you've got the crystallization fraction that could be attached to other cells. All right, so that's opsonization. Our antibodies are circulating, they recognize, they bind, and then this is where the macrophages come in. They bind to that fraction, says, oh, you're bound to something. I'm going to start the process of trying to destroy you. Second use is neutralization, and this is probably heard it in terms of COVID. All right, what we're trying to do is the antibodies try to bind and prevent the action of that foreign invader. In a lot of cases, we're talking about a toxin. Right? If you can get a lot of antibodies binding so that the toxin receptor site is blocked, then that toxin can't bind to its target. It's the same thing with the coronavirus that we're talking about. If you can get that virus encapsulated by antibodies so that the receptor or the binding site is blocked by the antibodies, you've effectively neutralized that pathogen. Now, IgM and IgG neutralize bacterial toxins. IgA also blocks toxins, but they're typically restricted to digestive and respiratory systems. All right. If they are just generic antibodies generic neutralizing antibodies, they're going to be a mix of the different types. All right, and they'll be a percentage. They'll say some of these are IgM, some of these are IgAs, and so forth. Third thing of antibodies, activation of complement. So complement's part of the innate immune response, also part of the memory response. This is just something complement says, hey, there is a foreign invader, turn me on so I can come in and start poking holes in the membranes and disrupt that cell. All right, and then our last thing is this antibody-dependent cell-mediated cytotoxicity. This is kind of like complement in its function. All right, it's going to bind, it's going to try to disrupt the cell membrane. All right, but these are antibodies that are going to bind to it and then call in the cells that antibodies, these eosinophils, neutrophils, lymphoid cells are going to come in and then bind to that antibody and try to poke the holes in the membranes. We'll see some of this antibody-dependent cell-mediated cytotoxicity. So the complement, not really effective, but this is. So that means we have to have developed a memory response first in order to exhibit this type of protection. This will kill the bloodstream form of Trypanosoma cruzi, so Chagas disease. We have that here in Texas. Right. So how does this work? This is rough overview of antigen presentation. So antigen comes in, gets engulfed, macrophages, gets processed onto the surface. That surface binds to T cells that then shuttle things off to form B cells. Okay? Forms B cells, and that starts to get our memory response started. That memory response, B cells, are going to new B cells, memory B cells, will have the antibodies on it. So if we see that again, we bind, we initiate the response, and then often leads to increased production of the antibodies and the B cells itself. What these B cells are going to do is present the antigen to the T cells and try to get things going. These are the T helper cells. Now, of the T cells, we've got T helper 1 and T helper 2 cells. So when I learned it, we were taught that it was you've got one side or the other. We're either going to go to a Th1 type response or a Th2 type response. Th1 is predominantly cell-mediated immunity. Th2 is primarily the humoral response or the antibody response. We've since learned that it's not necessarily isolated. It's not one or the other. It can be more of one than the other, but it's not going to be just entirely one or entirely the other. Now, what's interesting, if we predominantly go one direction, we tend to suppress the other side. So if a Th1 type response is really effective at killing off a parasite, what might the parasite try to do? Shift it the other way. Try to cause a switch that moves towards the Th2 type response, because if the Th2 type response gets stimulated, it's going to try to suppress the Th1, which allows the parasite to survive. We'll see that. We'll see some of that. All right. Other T cells, cytotoxic T lymphocytes. These are CD8 plus cells. They're going to kill our target cells that have these antigens on them. And then the Tregs, regulatory T cells, try to keep the whole thing under control. Because if we ramp up this response too much, you can kill it. We've talked about, you've probably heard like this cytokine storm. All right. In influenza, it seems like what kind of leads to some of this isn't necessarily the cytokine storm, but it's the regulatory T cells that can't keep things in check. Like somehow they get blocked, so then your immune system runs out of control. All right, so here's an example of it. You've got your Th1 arm, Th2 arm down here. This is the cell-mediated, not antibody-dependent. The humoral response, that is our antibodies and so forth. And you can see the interrelationship. You've got your uncommitted T cells that are going to ultimately develop as a Th1 or a Th2 type response. And then you've got all of these cytokines and chemokines that have the interplay. So if we're predominantly, let's say, Th1 type response, ways we can start to shift things over is to block, prevent expression, or bind up some of these chemokines or cytokines so that we don't have this inhibition. So you can see like interferon gamma stimulates macrophage development. If macrophages are really successful at clearing out the infection, what if we bind or inhibit this interferon gamma response? Well, this interferon gamma is also inhibiting this side. So if we block that, this side is no longer inhibited, we can shift our responses. All right? And same thing with the IL-4, IL-10. They're inhibiting Th1 type response. If we can stop those, if we can prevent those from acting, we freed up that inhibition and hopefully shifted things so that our parasite can survive. All right, so humoral response, our antigen presenting cells bind to Th2. All right, we're going to get proliferation of the B cells and so forth. We kind of talked about that, and the antibodies is providing all of these responses. The cell-mediated response is going to be predominantly this Th1. All right, the cells that are important are the natural killer cells, cytotoxic T lymphocytes, macrophages, granulocytes, and so forth. So that side was just kind of summarizing what I had mentioned. There are things, though, that we will see manifest in our host. All right. So one is delayed type hypersensitivity responses. All right. These are responses where the Th1 secretes cytokines that lead to inflammation, and that inflammation then draws in macrophages. All right. This could be a benefit for the parasite, especially if the parasite infects those macrophages. So it's like, hey, it's using our own body to get the cells that it needs. Cytolytic T lymphocyte responses. Now our cytotoxic T lymphocytes bind and inject perforin to perforate the target. All right, it's a little bit different than complement. Complement will cause these holes to appear, but it's not through perforin. Natural killer cells, completely non-specific. They're going to try to lyse the target cells. Again, they're not poking holes, they're trying to lyse. Now, the delayed type cytotoxic T lymphocytes, natural killer cells, they take time to develop. But there is an immediate hypersensitivity reaction that can happen, and it happens fast. This is the basis for the allergies, why some people carry these EpiPens around. They see it, and then all of a sudden, it is a very rapid onset of this inflammatory response. This response is dominated by this immunoglobulin epsilon, IgE, and the Th2 arm. All right, and this is pretty bad. And what's happening is the eosinophils get recruited. Towards the end of this, that you have this antibody-dependent cell cytotoxicity type reaction. What we're going to probably do is stop here and talk then about this inflammation, because that's really going to be a lot of pathology that we see. The parasite's in there, the host is fighting it. Well, it starts to lose, or just the initial response is, hey, let's increase inflammation. Let's get our effector cells there. What's the consequence of that? What's the consequence of it? Maybe, how does it manifest itself? So we'll finish this up on Monday. And then what we'll do is we'll start the actual content. Again, this is all FYI. We will see a lot of this stuff reappear, where we'll spend a little bit more time describing what's happening and how the parasite utilizes it to its advantage. On Blackboard, I posted some stuff. All right, so since this was FYI, you have the entire presentation, but the other presentations you don't get. So figures like this that I put up, I'm gonna have them into a separate handout document. Those handouts are posted. You can download them, you can work with them, you can take notes on them. And definitely you'll see that there's already, I think, 2 of them up. Third one probably get up on Monday, but they're actually here, copies, or at least one. So we start with platyhelminthes and go over. So I did the diagrams, copy and pasted it so you have it, so you don't have to frantically try to draw it out yourself. You've got it on here, take your notes. I got the spitibotherines posted as well. All right, so just look. All right, enjoy the weekend. First full week of classes, that means we have 14 weeks left? No spring break.